Okay, Chapter 10, Sensory Physiology. So in this chapter, we'll be talking about some general properties of the sensory system. We'll discuss our somatic senses and then our chemoreception, so the sense of smell and taste, and then we'll look at the ear, so hearing and equilibrium, and I believe we're also going to look at the eye. So general properties. So the sensory system is allows us to learn about what's going on in the body. And then our sensory neurons will bring that information into the central nervous system to then utilize that to figure out how we want to affect or what we want to tell our body to do. So sensory receptors are obviously going to be very important. Without understanding what's going on in our body, we won't be able to make the appropriate decisions. Now, sensory stimuli are divided into special senses and somatic senses. So some senses we are also conscious of and others we are not aware of. It's subconscious. So some special senses we are aware of include things like vision, hearing, taste, smell, equilibrium. And somatic senses that we are aware of include touch, temperature, pain, itch, proprioception. And then some subconscious senses include things like blood pressure. So, our body has to regulate blood pressure, and it can only do that if it knows what our blood pressure is. So we have baroreceptors that can do that, but it's not like you know what your blood pressure is. Now, you might feel the consequences of having very low or very high blood pressure, but it's not the same as our sense of pain. So here are some different types of sensory receptors. We have chemoreceptors, which sense chemicals like oxygen, hydrogen ions for pH, different molecules like glucose so that we can figure out our blood glucose levels. We have mechanoreceptors. They sense physical things like pressure, which are our baroreceptors, cell stretch, our osmoreceptors, vibration, acceleration, sound. Then photoreceptors sense photons of light, which helps us see, and then thermoreceptors sense different degrees of temperature. Now, one thing to understand about receptors is a receptor's receptive field. Okay, so the receptive field is the physical area, the field, to which a sensory neuron responds to stimuli. Okay, so if we take a look at this blue neuron, so this is a sensory neuron, this blue neuron will feel, okay, so let's imagine this is someone's arm, okay, so it will feel or it will respond, okay, or feel if you touch anywhere that is blue. So even though over here is pretty far away from the neuron compared to if I were touching right here, if I touched right here, our blue neuron wouldn't feel it. If I touched over here, it would. Why? Because over here is within the blue area. So the area is, this blue area is this blue neuron's receptive field. Now, just because I'm touching way over here, it doesn't mean I have to touch harder than if I were right next to the neuron. As long as I am touching anywhere in the receptive field, this neuron will respond as long as threshold is met. Now, the receptive field of a receptor can be irregular in shape, right? These look irregular to me. They can overlap. So you can see how we have overlapping. So if I were to touch right here, both the yellow and the purple neurons would respond. Now notice all three sensory neurons in the spinal cord, they are all converging onto the same secondary neuron. This secondary neuron happens to cross at the level of the spinal cord, and then it will send the information up to the brain to the appropriate area of the somatosensory cortex so that you can perceive the sensation. Now, another thing we can discuss about our receptors and receptive fields is two-point discrimination. Okay, so here we have a compass, equal distance apart in figure A and B. In figure A, we have two receptive fields being activated. Same thing in figure B. But in figure A, our brain will only feel one touch. In figure B, our brain will feel two touches. Why? Because all of these primary sensory neurons are converging onto the same secondary neuron. So this is an example of spatial summation. We talked about this when we talked about the neurons and how they work. This secondary neuron will kind of collect all of the sensations and then send up one signal to the brain. Over here, we have far less convergence happening. And so two separate secondary neurons are being activated. And as a result, we have two neurons sending a signal up to the brain. So we will actually be able to feel two pokes. Now, most of our cortical spinal tracts, so neurons that go from the brain to the spinal cord or from the spinal cord up to the brain, pass through the thalamus. So the thalamus is our relay station. You learn this in anatomy. It's going to relay the information to the appropriate area of, in this case, to the brain. The only exception for the sensory system is our sense of smell. So the olfactory neurons, they actually do not go to the thalamus. They go directly to the olfactory cortex. So that is an exception. Now, if we take a look at the somatosensory cortex, this is where we are able to perceive the sensation. And here's another way of looking at the somatosensory cortex. Now, what you see here is you see body parts lying over the somatosensory cortex. This is known as our homunculus. So basically, the body part that lies over the brain region, okay, they correlate with one another. So that means this area of my somatosensory cortex is going to feel from my hand. This area is going to feel from the arm, etc. What I want you to notice is the body parts are not proportional. For example, the hand is huge. Compare the hand to the entire torso, the entire back. So notice the back in real life is much larger than our hand, but in this figure, the hand looks much bigger. Why is that? Because our hand is much more important in touch, in sensation, to be able to distinguish what we are touching. We don't go around touching things with our back. We need to be able to feel on our back in case we feel pain or just kind of, general things. But it's not as important to be so particular in distinguishing touch. And as a result, we're going to dedicate more areas, a bigger area of our somatosensory cortex, to the areas of their body that are more important for touch in this case. So that's our homunculus. Okay, next. We know that the right side of the brain controls and feels from the left side of the body and vice versa. So 90% of those cortical spinal tracts cross at the medulla, the 10% cross at the level of the spinal cord. And the ones that cross at the level of the spinal cord are things like nociception, so pain fibers, temperature, coarse touch. And speaking of pain, so we have our nociceptors. So these are free nerve endings, and they're going to respond to pain, whether it's chemical, mechanical, or thermal stimuli. And nociceptors can activate two pathways. The first is our reflexive protective response. So it's a reflex. It's known as our withdrawal reflex, and it's integrated in the spinal cord. So the brain is actually not involved here. So think of if you've ever touched something hot. So let's say you touched a hot stove, your hand will move away right away, even before you actually perceive the pain. That's our reflexive protective response, our withdrawal reflex kicking in. We also have our ascending pathway. So what happens is when we touch that hot stove, we're going to move our hand away right away, even before we feel the pain, and then what happens? Then the pain comes on. That's because the ascending pathway eventually made its way to the somatosensory cortex, and then we sensed the pain. That's when the pain becomes, we become aware of it. Why do we need this withdrawal reflex? Because if we didn't have this reflexive protective response, it helps protect us. And if we only relied on our ascending pathway, that means when we touch something hot, like our hot stove, we're going to keep touching it until the ascending pathway makes its way to the somatosensory cortex to tell us it hurts. Then we have to send the impulse back down to tell our muscles to move our arm away. So having this withdrawal reflex that goes into the spinal cord and right back out is going to help protect us from pain or from injury due to that pain. One theory related to pain I want to go over is our gate control theory of pain. So the gate control theory of pain involves this little guy here in blue. This is our inhibitory inner neuron. So take a look. We're actually looking inside the spinal cord. So if you take a look at this rectangle, this is a blow up of it. We have certain fibers that are pain fibers. For example, this neuron that's coming in is a pain fiber. It's known as a slow pain fiber. So it's small in diameter. It is not myelinated, so action potentials travel slowly. Slow conduction velocity. This slow pain fiber, when it makes its way into the spinal cord, does two things. It stimulates this second pathway that will then go up to the brain to send the pain signal to the brain. But it can also innervate or stimulate this guy here. Now this guy is an inhibitory interneuron. So remember, an inner neuron is a neuron that's completely within the central nervous system between two neurons. But this guy is inhibitory. So inhibitory means it has an IPSP. So if you recall, IPSPs are going to decrease or hyperpolarize the membrane potential of the neuron that it's going to innervate, making this neuron less likely to fire. So this fiber, when it fires and goes up to the brain, it's basically sending a pain signal. We don't want this neuron to randomly fire, so we will have an inhibitory inner neuron inhibiting this neuron. Okay, so basically this neuron in the spinal cord is being inhibited. But now let's say a painful stimuli actually does occur. So here's our C fiber, our slow pain fiber, bringing that action potential into the spinal cord. It will do two things. It will stimulate our secondary neuron to send that pain signal up to the brain. But at the same time, it will inhibit the inhibitory inner neuron. If we didn't inhibit the inhibitory inner neuron, then we're going to have competition. We're going to have one IPSP and one EPSP, so nothing will go up to the brain. They'll cancel each other out, essentially. So by inhibiting the inhibitor, the inhibition goes away, so the entire pain signal can go up to the brain. So we know we got hurt. Now let's take a look at something called the gate control theory of pain modulation. Now we're going to use this to help modulate our pain. So what we're going to do is we have our painful stimuli. So there's our C fiber doing what we said. It's going to stimulate the secondary neuron while inhibiting the inhibitor. At the same time, we're going to touch the area. This is a non-painful stimuli, so it's going to activate different neurons. This is an A beta fiber. So it's going to bring the stimuli into the spinal cord, but because we're touching the same area in a non-painful way, what this fiber is going to do is it's going to stimulate the inhibitor a little bit. So here we're inhibiting, here we are stimulating, and so what happens is a little bit of the inhibition remains. So this inhibitory inner neuron somewhat inhibits the secondary neuron. So notice the color is much lighter. And so the pain signal that goes up to the brain is not going to be as intense. So here we are modulating the pain using this gate. That inhibitory inner neuron is our gate. Another theory related to pain is referred pain. A referred pain is the best example, or the classic example, is when you have a heart attack and then you have pain along your left arm. So referred pain really occurs because neurons from different parts, from an internal organ in this example, the kidneys, and an area of the skin, these two sensory neurons actually converge onto the same secondary neuron. So when you actually have pain in the kidney due to a kidney stone, let's say, or we've destroyed our nephrons for some reason, high blood pressure, or infection, whatever it is, the pain signal that goes into the spinal cord is going to innervate the same neuron that would normally be activated by this area of the skin. And so when the pain signal goes up to the brain, it's going to send the pain signal up to the area of the somatosensory cortex associated with this area of the skin. And so this is why when you have pain in the kidneys, this area of the skin hurts. Now in general, the area of the skin kind of matches where the organ is located. So here's your heart, and so a heart attack kind of pain radiates this way. Here is your intestine, And as a result, the pain radiates where the intestines are located, or the stomach, or the colon, or the appendix. So it's within the same region typically. Sometimes it's not. For example, here we have the liver and gallbladder. You're going to have pain up here in your upper right shoulder. But it really isn't about where the organ is located. It's about which neurons are converging. Okay, so let's move on to our special senses. Let's start with olfaction, our sense of smell. So the olfactory system helps us smell. What was unique about it? These neurons don't go to the thalamus. They go directly to the olfactory cortex. They also regenerate, and they're involved in smell. And we know smell is linked to things like our memory and emotion. So that's pretty much it for olfaction. The next special sense is our sense of taste, or our gustatory sense. And we have 5 taste buds. We have bitter, sweet, umami, sour, and salty. So I'm sure we're all familiar with salty and sour, sweet and bitter, but what the heck is umami? So umami is activated by things like certain amino acids, glutamate, nucleotides, and it creates that taste of something tasting really savory, delicious, nutritious. It doesn't necessarily mean it is nutritious, but that's how it can taste. So these are our different senses. Now, receptors, even when you activate receptors, that's not enough. Remember, the signal has to make its way into our brain. So obviously, once we've activated the receptors, it has to transduce the signal into an action potential. And so we're looking at that happening here. So here's, our voltage-gated calcium channel opening, calcium coming in. We're releasing neurotransmitters. That's activating a neuron, and then the neuron will send that impulse, that signal, through an action potential, or as an action potential, to the brain so that we can interpret or perceive this taste. And we can even develop special tastes, like sometimes you really want something salty or something sweet. We can also train our taste buds. If you're always eating something sweet, you can kind of cut it out of your diet for a while, and then you won't need something as sweet to actually enjoy the sweetness. Okay, next we have our ear, which is going to help us hear, and it's also involved in equilibrium. So the anatomy of the ear is something you should already be familiar with. And here what we're seeing is our sound waves. It's coming in through our outer ear. It's hitting the tympanic membrane, and all the little bones inside are vibrating. So it's sending the sound into the ear through the vibrations into our inner ear. Now, the receptors that actually respond to sound or help us hear, are our hair cells. So our hair cells are shown here and over here. They have a stereocilia kind of shape, which basically means it has these kind of fingers or hairs that go from short to taller and taller. And notice each hair cell has a neuron below it. So remember, it's not about just activating the receptor. The receptor then has to send the signal through the nervous system to our brain. So that's why we have to have these neurons. Now, within our inner ear, we have all of our hair cells, and we also have this basilar membrane. Now, when the vibrations come in through our middle, into our inner ear, that vibration is going to cause this basilar membrane to vibrate, to shake. Now, depending on the pitch, different areas will vibrate. If it's high pitch, high frequency, the area of the basilar membrane that is closer to the oval window, that is stiffer, is going to vibrate. It's thicker and stiffer. If it's low pitch or low frequency sound, then the area further away, that is flatter and thinner and more flexible, is going to vibrate. And so depending on which area vibrates, those hair cells will be activated, and that's how we hear. And again, another function of the ear is to help with equilibrium, as well as movement and our position in space. And it'll work in conjunction with the cerebellum. All right, and finally we have our eye. So the eye obviously is important for vision, and light enters the eye and it gets focused on the retina, which is in the back of the eye. Photoreceptors are on the retina, and those are the receptors that sense photons of light. So let's take a look. So here we have our cornea. Here's our pupil. That's that black area in our eye that constricts or dilates, depending on whether we're outside in the sunlight or inside where it's darker. So that tells you the pupils control the amount of light that enters the eye. So when people turn off the lights, we can't see anything initially, and then we say our eyes adjust, and then we can start to see. So our eyes adjusting is our pupil dilating, so that more light can be brought in. Then we have our lens. The lens helps us bend the light so that the light rays that come into our eye will converge, come together to a focal point. And what we want is we want that focal point to land on the retina. And the retina is way back here. And this is where we have our photoreceptors, so the receptors that will actually sense the photons of light. Ciliary muscles helps contraction, or when they contract, they help alter the curvature of the lens. What else do I want to point out? The fovea. The fovea is the area of sharpest vision. And I'll talk about why that is later. And then we also have the optic disc. It's shown over here as well. This is also known as your blind spot, and this is where you actually have no photoreceptors. Instead, this is where your blood vessels and your nerve vessels will enter and exit the eye. So no photoreceptors here. And what else? Let's move on here then. Okay, so about bending light. So we have different types of lenses that we could use to help with bending light. So we have concave and convex. A concave lens, what it does is light rays come in, and notice it scatters the light. So when we use a concave lens, what that's going to do is it's going to scatter the light and move the focal point further back. A convex lens, notice, what does it do? It causes the light rays to converge, to come together sooner. And so what it does is it causes our focal point to become shorter, or the focal length to become shorter. So our focal point comes closer. So instead of moving further back, it comes closer. And so depending on whether we're myopic or hyperopic, we'll use the appropriate lens. So let's start with hyperopia. So someone who is hyperopic, you are far-sighted. And so what happens here is when the light rays come in, and our retina is right here, this first black line that you see, we want the focal point of our light rays to hit the retina. In farsightedness, hyperopia, it's landing behind the retina. So what we need is the light rays to come together sooner. We need this focal point to move back a little bit. So we need this focal length to shorten. So we're going to use a convex lens. And notice that convex lens brings the focal point to the retina. If you have myopia or you're nearsighted, the focal point lands before the retina. And so you need a concave lens to scatter the light a little bit to move the focal point back. And if we take a closer look at the retina, here are our photoreceptors. Okay, so these things in purple and yellow are photoreceptors. And we have different types of photoreceptors. We have our rods and our cones. Our rods are involved in low light, night vision, black and white vision, and they're shown in yellow. So notice we have a lot more rods than cones. Our cones are our high acuity vision. That's for our color vision. During the day, we have our blue cones, our green cones, and our red cones. Now, what I want you to notice is the light's coming in this way. Okay, so the lens and everything, the pupil is over here. Light's coming in this way. We want the light rays to hit our retina, which is way back here. Notice we have all of this other stuff in front of the retina. These are our ganglia cells. Now, why is the fovea the area of sharpest vision? Sorry, this says fovea. Why is that the area of sharpest vision? Because the fovea, notice the ganglia cells are moved out of the way. So the light rays, they don't have to kind of make its way through here to hit the photoreceptors. It will directly hit the photoreceptors. So that's one reason. The other reason is, if we look at the retina anywhere else, notice we have a lot more rods than cones. But at the fovea, it's all cones, which is our high acuity color vision. And so this makes our fovea the area of sharpest vision. And that is it for this chapter. Thank you.